So yes, I'm Lyndle. I'm a postdoctoral researcher employed by Curtin University, but based on the other side of the country at the Australian Plant Bank that we've heard quite a lot about today. So the Botanic Gardens of Sydney, when we speak about it, is not just the Botanic Garden based in the centre of the city. It encompasses 3 different botanic gardens around the greater Sydney region. They're based on Gadigal country, on Darug country, and on Darawal country. And so the Australian Plant Bank is on Darawal country, and I'd like to acknowledge that that is where I both live and work and continue to learn. And so I'd like to acknowledge the elders for their ongoing custodianship of that land and for allowing me to be a part of that custodianship, and acknowledge that sovereignty was never ceded. So this artwork here is by Darren, and he tried to sort of encompass that connection to land and connection between different countries, and particularly the 3 botanic gardens. And I just think it's a really beautiful representation of those connections. So these are the 3 botanic gardens that we've got. So Sydney, based right in the centre of the city. Garden just turned 209 last week. So it's a very small garden, but it has plants from all over the world, and so it's really quite spectacular. Obviously, got views of the harbour. We then got the Australian Botanic Gardens out at Mount Allen, so we're nearly 60 kilometres from the centre of the city, but still part of that Sydney region. Very different climate compared to what's in the centre of the city, and we've only got Australian flora. So it's a much larger area, but plants from all over the country. That is also where our scientific research hubs are. So that's where the National Herbarium is and where the Australian Plant Bank is. And then our final botanic garden, another small one in the Blue Mountains. Again, plants from all over the world, but because of where it's located, much more cool climate plants, but they also have a huge amount of natural environment there as well. And I'd like to acknowledge before I continue that the work that I'm doing, or that I'm speaking about today, is not just my own work, it's work from all different teams across the Botanic Gardens. So hopefully they'll forgive me if I misrepresent anything. So huge collections across the 3 Botanic Gardens. Garden specimens, so these are the plants that are growing out in the garden for display. Over 1,000 different species, cultivars, and varieties. And that distribution between species is quite dependent on what genera plants come from. So things like eucalyptus and other gum trees, lots of different species, also for the syzygiums or the lily-billy relatives. Whereas for things like olive and tea trees, it's more of those varieties, those different cultivars that grow in people's gardens. They've got a lot of those, but not so many individual species. In tissue culture, we have 15 Myrtle species. Some of those we just have one individual represented in the collection. Other ones, particularly the Myrtle rust affected species that we're focused on at the moment, we have lots of different accessions, so lots of individuals, particularly of Rhodomonas sidioides, the native guava, Rhodamia rubescens, or the scrub turpentine, and Syzygium paniculatum. And we've got the seed bank, who stores seeds from lots of different Myrtle species, subspecies, and crosses. And once again, there's quite a lot of diversity in how big those collections are. Some of those collections, it might be one collection from one individual with just a few seeds. Other times, it's thousands of seeds from different individuals from all over the country. So those numbers are very much simplifying what is there, but it is large collections. So we get plant material from all sorts of different people. It comes in different forms. It could be cuttings. You can see some of Craig's cuttings up the top there. We can get fruit. We might get seeds. And what we do with those plant material is really dependent on the quality, what type of material it is, how it's going to need to be moved around, how much of it we can get, and then what that end purpose is. So whether it is to plant it out in the gardens, or whether the idea is to do research on it, whether it's a backup collection for another organisation or community group. So all those things will affect the way that we manage that material within the gardens. And I've listed storage potential there. So coming back to your question about seed banking, some seeds can be stored, they can be dried down, they can be frozen, stuck in the seed bank. We call those orthodox seeds. They're great to store away. Unfortunately, a lot of species are not orthodox, and so they might have desiccation sensitivities, or they might have freezing sensitivities. They might be short-lived in storage, dormancy issues that mean that they're really hard to store because we don't know how to get them to grow after. So as an example from the species that we've heard about, Rhodamia rubescens is an orthodox species, so it goes really well in the seed bank when we can get the seeds to put in the seed bank. But Rhodomonas stibioides is a freezing sensitive species. So we can dry it down to some degree, but once we stick it in the freezer, it doesn't live for very long. And so we're doing a lot of research to work out how we deal with those species. Lily pillies, you can't dry them out at all. Very upset when they get dried. So there's other work that we have to do to manage those plants as they come in. And then obviously we have those limitations that other people have spoken about. So do we have enough space for the material that's coming? Do we have enough people to work on it? Do we have the funding to keep it going? I wanted to talk a little bit about how plants are moved around the gardens and all the different groups that work on it. So we can get plants from the wild. Things might end up in the seed bank. They might end up in the nursery, growing out for display. They can be used for research. They might end up at other organisations and used in cryo storage and in tissue culture. And I'm about to make this diagram confusing. Hope you're ready. So plants from the wild can end up in any of these places. They'll be used for different purposes. Seed bank will send material to the nursery. They'll send material to other seed banks, other collections. They'll send material for research. And that research can be things like doing genetic analysis. It might be those resistance assays. It might be metabolomics. It could be anything that other people are interested in. It might go to students. It might be internal research. Could be anything. Nursery is sending material out to everyone. The gardens will send material back to the nursery to be propagated. If a plant is starting to look old in the garden, it might be come back to the nursery to sort of be spruced up a bit before it gets sent out. We'll also collect material from the gardens to put it into tissue culture. From tissue culture, material goes back to the nursery to put on roots and to grow out in the wild. It might be sent for research, goes to external collections, so we send plants up to UQ all the time. Material comes from cryopreservation back out to tissue culture, and some external collections they'll also send us material. As you can see, there is a lot of movement. We've got a lot of collaboration between different teams, and you might notice there that the core nursery has a lot of teams coming and going all the time. So they have a very tiny but very mighty team led by Ryan, and we also have our research horticulturists who work in the nursery as well, mainly on those lost collections. So it's a bit of a bottleneck, and we do need to sort of communicate with each other about, well, if I bring in material now, is there space for it? Is there someone to work on it? What do we need to remove from the nursery to make space for the new things? And that's kind of a consideration across all these groups. We've heard from Craig already a bit about how we're trying to maximize those collections that we do have. So with limited amount of space, we want to make sure that we've got the best quality material that we can have. So genetics has been really important in that. And so I'd like to acknowledge the work that the RESA team does, one of our Botanic Gardens teams in the city. So they do a lot of this genetic work, and they've determined that those collections that we're holding for Rhodomonas and Rhodamia are representative of the wild populations. So that the material has come from across the distribution, but it is also genetically representative. And we've also found through those Myrtle rust assays that we have plants that are highly susceptible. We have plants that are moderately susceptible. We have plants that are more tolerant. So that's been really important for us to make sure we have that diversity within our collections. But what do we do when we run out of space? It's all very well to have all of this stuff, but we can't manage it for every species. We've got another 22 species out there, but not much has been done on how do we manage all of this. And that's where cryo technology comes in. So we've already heard a little bit about the tissue culture and cryo storage that's going on at UQ, and that is the work that I do. So tissue culture, we can store the plants in these jars. They've got jelly that contains everything they need to grow, and it buys us some time while other people are working on their different solutions. So we can keep those plants in a really controlled environment, away from disease, away from the environmental stresses, and keep them safe while we work on all of this other stuff. But as I said, we can use it for all sorts of different experiments as well. So this material gets used in all sorts of different things. So some of those collections that we maintain are quite large to allow us to use the material in so many different ways. And then cryo, things are stored in liquid nitrogen, so it's preventing those metabolic processes from going on. We can store huge amounts of material, of things that are very small, and it allows us to store a lot more in a much smaller space. So in essence, it can increase the biodiversity that we're holding, but without all of that ongoing maintenance that those other collections require. However, like many things, the success of the cryo is going to depend on the quality of the material that we have. So when I was doing the PhD research, I was trying to focus on seed material, thinking, well, seeds have much better biodiversity within them. There's a lot more genetic diversity in seeds than there are in clonal shoot tips. But I would receive lily pillies that looked like this. They had been damaged so much by the insects that I was getting so much contamination, I wasn't getting anything surviving. Or when I was working on aniseed myrtle, which is this X-ray image you can see, there's only 2 seeds in that image. The rest of it is resin inside an empty seed capsule. And so that particular year, I had less than 0.5% of all the fruit I collected had a seed inside. So you can imagine the amount of work that goes into that to try and find a viable seed. There is no point banking something or sticking something in cryo that is not going to grow. So that was quite a difficult process, and that's why I moved on to using shoot tips. But it's quite difficult to develop these processes, and you've heard some of that process from Jingyin and Van Ann this morning. These are examples of 2 rhodomonas shoot tips that I was working with. The top is the control. It was just cut off and put on to new media. It took it 2 months to put out those 2 leaves, and it never grew any further. But it looks great compared to the one below that was put through cryo storage. And you can only just see it, but it hasn't put out new leaves. But it took it 3 months to produce those 2 leaves, and it never progressed any further. And so there's still a lot of work to be done optimizing those procedures so that we get better results, better recovery. So what can we do to do that? We've heard from these guys this morning about how we can optimize the media. So these are the main elements that are found in that tissue culture media. You might recognize a lot of those words. They're vitamins that we as humans need to grow as well. But those are all the things that would be found in the soil that the plants are taking up to help them build and grow. So we thought, well, what happens if we mess around with the amounts of those things? Can I get the plants to grow better? Thinking about the fact that, you know, as humans, when we're babies, we have very different nutritional needs to the fact to when we're adults or to when we're older in life. Different life stages, we have different requirements. So maybe those shoot tips are not doing as well on the media I'm using for small plants. So maybe that's what the issue is. So I set up a huge experiment with 460 shoot tips, and every 4 weeks I measured how long they were, how many leaves they had, how healthy they looked, what color they were, and I did that for 3 different species. So I've done it for Syzygium paniculatum on the lily pilly. I've done it for Rhodomonas sidioides. That one is still in the lab, so this was a photo taken last week. But you can see even after 6 weeks, there's quite a lot of variation between the different medias that the plants are growing on. So there's some that haven't grown at all. There's others that have put out 1 or 2 leaves, and then there are others that are already starting to multiply within the jar. And then we had our Rhodomia rubescens, which after 16 weeks had grown from 1.5 millimeters tall to these 4 centimeter tall plants with lots of shoots and lots of leaves and everything else. Starting to decline because it's no longer a media that's good for it. So it's great for it to grow from that shoot tip size to get a little bit bigger, but it's starting to struggle now. So then I moved it onto the regular media and it's doing quite well. Oh, I need to speed up. So these are the different medias that I looked at. This is the control media. This is the media that we typically grow them on. And then we've got the top performing media that I tested for 2 different species. And you can see there's a lot of variation in those. So there's not one true answer to what those shoot tips need to grow. But overall, I did find that they needed extra iron. They needed extra microelements. I'm not sure which of the microelements they wanted, but they did want at least one of them. And I found that they needed less nitrates. And differently to what Jean found for the Gossyas, mine didn't like the ammonium nitrate, whereas her species wanted more of that ammonium nitrate. So there's quite a difference between species. And that's one of the difficulties of working with cryo and tissue culture. In many cases, it is species dependent and sometimes even individual dependent. So where do I go to next? I'm going to continue working on the cryo storage protocols, but using those optimized media to try and get better results in cryo. So I'm hoping to get the same results as these guys have had using that optimized media to get better results from cryo, but also looking at some of those other steps. I've been using some other tools just to get a better idea of what's going on in that plant material. But the idea is that we're moving from these collections that we have in so many different places in the garden that are such high maintenance collections, so valuable but take a lot of work to maintain, to these lower maintenance collections. We can stick them in a cryo vessel and leave them for a bit while we work on other species and other things. So yeah, that's where we're headed. I'd like to acknowledge that Curtin Medical School has paid for me to be here today and the funding that comes from different groups. And then my colleagues at the different universities and different organisations will just report the changes into me as well. So thank you.